Students, a very warm welcome to our phycology class. The topic for today's discussion is thallus organization in algae or range of thallus organization in algal cells. Okay, you can call it by any name. Now see, here we are going to study the different types of thallus found in different algal communities, right? The thallus organization seen in different class of algae shows that there is progressive evolution in their structure from a unicellular simple form to a more complex multicellular form, right? So let's get started. See, this flow chart gives you a vivid idea about the different types of thallus that you get, right? See, initially, they can be classified under two major subheads. One is unicellular, the other is multicellular. Okay? In unicellular, under unicellular, you see, you will get motile colonies and you will get motile organisms and you will get non-motile organism. Motile organism means what? They are capable of movement, and non-motile organisms are those which are incapable of movement, right? Next we have multicellular. This subhead, this category, multicellular, which can again be classified into two types, non-motile colony and motile colony, right? Since they are multicellular, they organize themselves in colonies. Here you will not find because they are unicellular. Fine. Motile colony can again be subdivided into amorphous colony and dendroid colony. Fine. This is microcystis, M-I-C-R-O, C-Y-S-T-I-S, okay? Microcystis. Examples are also given. I hope you are noting them down. See, in each box, under each category, the respective examples are also provided. Fine. See, this motile colony can be amorphous, dendroid, filamentous, and filamentous, heterotryphus. Please note them. See, amorphous, dendroid, filamentous, heterotryphus, Typhoonus, Pseudoparenchymatous, and Parenchymatous. Okay. Now this filamentous form can again be subdivided into unbranched, simple, that is simple branched, and pseudo-branched. Okay. Now we'll talk about each of these one by one. We will start with the unicellular group. What did we discuss just now? If we first take unicellular, right? Unicellular can be divided into non-motile and motile. Okay. In non-motile unicellular form, what do you see? A single non-motile cell carry out all essential functions of life. What do you see here? A single non-motile cell carry out all essential functions of life. Functions of life. That means what? They are so very independent. They themselves can perform everything required to sustain their life. Okay. They show many morphological variations in Bacillariophyceae, Chlorophyceae, Rhodophyceae. Okay. We will study each of these types with different examples. Examples, what do we see? Chlorella. Okay. Next, what do we have? This was the non-motile form, and now we'll talk about the motile form. Here, what do we see? A single cell. See, one thing you must note, non-motile means what? Here, it is very important to mention, flagella are absent. This is what is making them non-motile, isn't it? Absence of flagellum is making them non-motile. Now coming to the motile form. A single cell with flagella. Here this category includes single cell with flagella. Okay. Single cell with flagella for locomotion. Correct? This group varies greatly in size and shape. Okay. The unicellular forms may be spherical, oblong, pear-shaped, or sometimes elongated, bearing flagella. Right? If we quote an example here, the best suited example is Chlamydomonas. All right. Please try to remember the examples. Thallus organization in algae cannot be studied without example, isn't it? See, this motile forms, if we take a little deeper look into it, the motile forms can also be categorized into two types. The motile form can also be categorized into two types. What are they? One is flagellated. The other is rhizopodial. One is flagellated, the other is rhizopodial. Okay. Flagellated, see, this form, this flagellated form is found in major groups of algae except for Phaeophyceae, Rhodophyceae, Bacillariophyceae, and Myxophyceae. Okay. The distinguishing features are the presence of a unicellular plant body bearing flagella. Correct. The thallus is round, pear-shaped, as I have told you, and They bear two motile flagella at the anterior region. This is important to note. Flagella at the anterior position. Anterior position. Okay. Examples you have seen, it is Chlamydomonas. See, both the flagella may be equal in chlorophycy. When it's equal, it is in equal length flagella are found in chlorophycy. And when they are unequal, this type is found in xanthophycy or dinophycy. Okay. Now let me give you some examples of unicellular motile algae. Euglena is unicellular motile algae. Then you have Chlamydomonas, which I've already written. Then Cryptomonas, Cryptomonas, Cryptomonas. So try to remember some of these names for examples. All right. Next, we come to the rhizopodial form, rhizopodial type. Here, what do we see? The rhizopodial unicellular thalli have a naked protoplast without rigid cell wall. Here, the cell is bound by a rigid cell wall. In flagellated form, cell is bound by a rigid cell wall. But here, cell is naked. That means what? It is not bound by a rigid cell wall. Okay. The plant body here contains cytoplasmic projections called pseudopodia. Where have you heard of this word pseudopodia? In amoeba, isn't it? In amoeboid movement. So these projections are called pseudopodia and rhizopodia, and the flagella are completely absent. Okay, these organisms show amoeboid movement for locomotion. That is movement by means of cytoplasmic projections, movement by means of cytoplasmic projections, correct, called pseudopodia or rhizopodia. Pseudopodia and rhizopodia. Okay, examples of this type, that is, examples of rhizopodial forms are chrysamoeba of chrysophycy. Example I'm writing here. CH. Please don't make a mistake while writing the spellings. All right. So what did we learn over here? See, quickly take a note. We had started our discussion with unicellular form. Unicellular form can be divided broadly into two categories. One is the non-motile type and the other is the motile type. In non-motile type, you see here a single cell, a single non-motile cell. They perform every essential functions required. Example is chlorella. Okay, here flagella are absent. This is what makes them non-motile. In motile form, what do we see? Here, the single cell with flagella, the single cell is capable of movement by virtue of flagella. right? Suitable example is Chlamydomonas. Now this motile form can further be categorized into two types, flagellated and rhizopodial. All right, flagellated form and rhizopodial form. In flagellated form, you see flagella at the anterior position, that is, and the cell. Generally, the flagella is present at the anterior position and the cell is bound by a rigid cell wall. In rhizopodial form, what do you see? The protoplast or the cell is naked and the movement is carried out by means of cytoplasmic projections. This is very much similar to movements in amoeba, that is amoeboid movement. Amoeboid movement. All right. I hope till here it is clear. Now we'll move on to the multicellular form. Okay. In multicellular form, the colony is formed by aggregation of individual cells. Next, what do we have? The multicellular form. Here, the colony is formed by the aggregation of individual cells, aggregation of individual cells. All right. First, what do we have? We have non-motile colony. Please listen to this lecture with a pen and paper in your hand. Keep on noting these down. Whatever I'm writing here, please note them down carefully. Here, what do we see? Individual cells in the colony lack flagella and are often found to occur in synobium. Okay, individual cells. For your benefit, I'm writing each and every word that I'm saying. So please take them down. Individual cell in the colony lacks what? Flagella. Lacks flagella and are often found to occur in synobium. Now, what is a synobium? And are often found to occur in synobium. Often found to occur in synobium. All right. What is synobium? Synobium is a colony with definite number of cells and having a contrast shape and size. Okay, they have definite number of cells. Right? Examples, what do we have? We have pediastrum. All right. We also have synodesmus. All right. Next, we'll talk about motile colony. So, non-motile colony is clear. Next, we'll move on to motile colony. In multicellular, please don't get confused. In multicellular form, non-motile colony was there. Now we will talk about motile colony. Okay. What do we have here? Individual cells in the motile colony possess flagella for locomotion. Often a synobium is found to occur. All right, they are found to possess flagella. Obviously, we are calling them motile. That means what? They will have flagella for movement. Okay. So, individual cell in the colony possesses individual cell in the possesses what? Flagella. All right. Flagella for locomotion. See, in volvox, about 500 to 50,000 cells are interconnected with each other with division of labor. Okay. So example, what we can say, volvox, then gonium, all these we can quote as examples. Fine. Now you see in motile multicellular colony, we have different forms available. What are they? First one to be brought into discussion is Pomeloyd or amorphous colony. What is that? Pomeloyd or amorphous colony. What do we see here? Here we see numerous non-motile cells that remain embedded in a common mucilaginous matrix. They look like this. See. Suppose this is a mucilaginous matrix, right? Many cells, they remain embedded in the mucilaginous matrix. This is how it looks. Okay, this dotted portion is the mucilaginous matrix. Mucilaginous matrix. Okay. See, numerous non-motile cells are embedded in a common mucilaginous matrix. All right. Every time you do these detailed study, this chart in mind. So far, what have we covered? We had talked about this unicellular type, both we have covered here. Now we are talking about this multicellular form. In this multicellular form, we had already talked about this non-motile colony, motile colony. Now we are talking about amorphous colony. Okay, like this we will proceed. So what do we see here? Numerous non-motile cells are embedded in a common mucilaginous matrix. Okay. Here, all the cells are independent and fulfills all functions of an individual. Please remember, these cells are all independent. Okay. Example, you can write Pamela. You can also write Tetraspora. Okay. Next, what do we have? This was your C. This is your D. Next, what do we have? Dendroid form. Dendroid form. In dendroid form, what do we see? The plant body possesses a tree-like structure. Plant body possesses a tree-like structure. Okay. Mucilage is often found to occur at the base of the structure. Right? Most often the tree-like structure is like this. Suppose there is a tree-like structure. Okay, like this. Example, Dinobryon. Example, Dinobryon. Okay. Then what do we have? We have Filamentous form. Filamentous form. Here, see, the cells are arranged one upon the other. Cells are arranged one upon the other in a definite sequence or uniseriate rows forming filament. Let's write or uniseriate rows forming filament. Okay, filaments may be branched or unbranched. This can be again of two types. What is that? Branched filament and unbranched filament. Okay. Now see, as we study the branched and unbranched filaments, we will quote examples. We'll quote suitable examples. In branched filaments, the branching can often be dichotomous. What is dichotomous branching? Like this. Branching can often be dichotomous. Okay. Examples of branched filament. What do we get? Cladophora, Pithophora. All these are examples of branched filaments. Cladophora, Pithophora, anything you can write. Coming to unbranched, the cells are arranged one above the other without any branching. Like the unbranched filament will look like what? like this. The cells are arranged one above the other without any branching. Like this. See, these are the junctions. See, the cells are arranged one above the other, but there is no branching. Okay. Example, what? Spirogyra. Heard of this many times before. Then nostoc. How does nostoc look? They look like this, no? Beads in a chain. Nostoc looks like this. You can also write zygnema, anabina, all these, right? Then we'll come to pseudo. See, in filamentous form, we had talked about branched, unbranched. Other two types are pseudobranches and heterotryphus form. Okay, pseudobranches and heterotryphus form. Go to the fresh slide. See, pseudobranch type, what do we see? In pseudobranch type, we see, I'm again going back to this. These are the various, and this is what we are studying. We had talked about unbranched. We had talked about branched. Now pseudobranch. Then we'll talk about these four remaining: Heterotryphus, Typhoonus, Pseudoparenchymatous, and Parenchymatous. Okay. Heterotryphus can also be a part of this filament branching type also. Okay. See, in pseudobranch, what do we see? Pseudobranch condition, see what do we have here? They appear like branched under microscope. Pseudobranch, they appear like branch under microscope. This branching is due to close association of unbranched individuals. So what is important to note is branching is due to close association of unbranched individuals, of unbranched individuals, or individual filaments, not individuals, individual filaments, like you see in cytonema. Example, cytonema. Here what happens, you see, suppose it is somewhat like this. Then you have another branch like this. It is so closely associated with one another, like, see, as if this has got branched. As if this has got branched. Okay. So under microscope, it appears like branched ones, but this branching is not true branching. That is why it is called pseudobranching. Then what do we have? We have heterotryphus form. Heterotrychus form. What is that? Hetero means what? Different. Trichous means trichome or filament. Okay. So what do we have here? Presence of more than one type of filament. Hetero means different. So presence of different types of filament. What do we see here? Presence of more than one type of filament. Okay. Plant body consists of a prostrate system from which an erect system develops. This is also called primary projecting system. The branches may give rise to primary projecting system, secondary or tertiary projecting systems. Okay. Sometimes you will see many are occurring. Most famous examples, most well-known examples are ectocarpus. Right? Then we'll talk about the Typhoonus form. Okay. Typhoonus form. What is that? The plant body is elongated. Okay, the plant body is elongated without any septation. That means what? Septa formation hasn't taken place. Many nuclei are present in a common protoplasm. That means what? This means it is implied that many nuclei are present in a common protoplasm. All right. This is also known as Xenocytic condition. You must have heard of this word. Xenocytic condition. Okay. Example, Vaucaria. Other examples can be Botrydium also. Next, we have Pseudoparenchymatous form. Okay. Next, we have Pseudoparenchymatous form. In Pseudoparenchymatous, what do we have? We see close juxtaposition of branched system of a single main axial thread to form a thallus. What did I say? I repeat once again. Close juxtaposition. That means what? Close placement. They are closely placed. Close juxtaposition of branched system of a single main axial thread is seen to form a thallus. Okay. The plant body consists of one main pseudoparenchymatous axis and all other are side branches. Fine. All others are side branches. Clear? So let's write close juxtaposition of branched system of a branch system of a single main axial thread. Main axial thread is found to form a thallus. Is found to form a thallus, right? So what is it actually? The plant body consists of one main pseudoparenchymatous axis and all others are side branches, correct? All others are side branches. This can be uniaxial type, multi-axial type, okay? Next, we'll go to the parenchymatous form. Okay, let's, I've forgotten to add one example here. This is what? Monostroma. This can be one example. Monostroma. Next, we go to parenchymatous. Pseudo is over, parenchymatous form. Here, what do we see? The parenchymatous form of the plant body is a modification of the filamentous habit. Fine. The formation of a parenchymatous thallus occurs. Okay. Formation occurs, just writing in short. Formation occurs when cells of the primary filament, primary filament divide into two or more planes. Divide into two or more planes. Okay. The parenchymatous organization may be folios. This can be organization may be folios. Example, vulva. It can be tubular, right? Tubular, example, enteromorpha. Then it can be a highly developed structure. Highly developed structure. What is that? You know many of them. Cara, fucus, laminaria. Just write one. Okay, so this is how we come to an end of our discussion today. I hope you have understood and have simultaneously enjoyed learning this new lesson. In case you have any confusion anywhere, please feel free to get back to me in the comment section. Till then, thank you so much for joining with me. Goodbye.